Welcome to the webinar, and today I'm going to tell you about some of the development of this new technology that my lab has built on top of Mission Bio's Tapestri Platform, where really the objective of this technology is to enable us to start to actually link together genotypes and phenotypes at the single cell level at very high throughput. Next slide, Ben. So before we get into that, I just wanted to give everybody a little bit of background on the development of the technology and Mission Bio. So my lab at UCSF, we are a technology lab. My training is in physics, and what my lab focuses on is building instrumentation basically for biologists to do fundamental measurements. And we focus a lot on single cell analysis and a variety of other technologies that, that we are building currently. Next slide. So the development of the technology that became Mission Bio started back in 2013, and it really was recognizing that there is quite a significant need, which is that in order to do very sensitive measurements on single cells, especially to analyze the genomes of single cells, there needs to be a very robust way to lyse the cell and access the cellular genome. And so we spent actually several years developing technologies for very efficiently lysing cells in droplets on the order of 40 picoliters in volume that would then allow you to do an efficient PCR reaction. That technology, or a series of those technologies, essentially were published in sequences you can see here, which really demonstrated that we solved that major challenge of how do you get efficient molecular biology to happen on a single cell's genome. Next slide. So that technology then, once we had it working, became basically the foundation of Mission Bio. So Mission Bio packaged it into a really beautiful, user-friendly platform, which is now commercially available. And it essentially incorporates the essential steps of the workflow, the microfluidic workflow that we developed in the lab for efficiently lysing single cells and then doing a PCR off of the genome, where you could get a high probability of amplifying the target loci of interest. Of course, now Mission Bio offers this. You can do hundreds of loci simultaneously. The PCRs are quite uniform and efficient, and it works great. So this is an excellent technology for mapping genotypes when you have a very heterogeneous system, such as in cancer. And it allows you to really get to track the evolution of how the disease develops over time if you can do time-dependent measurements, and we'll talk a little bit about that in the following slides. Next slide. So, in a very simple way, doing genotyping is great for tracking the genome, of course, but there's going to be scenarios where you also want to understand something about the phenotypes of cells. And that information is not necessarily easy to get out of just a genotyping kind of measurement. So this was an idea that we had actually back in 2013 or 2014, where we thought, is there a way that we can somehow integrate phenotype information into the genotype measurements that we could perform with this technology? And there's essentially two ways of phenotyping in some sense. I mean, of course, it's very simplistic, but you can think of RNA-seq as being a coarse kind of phenotype, where you can look at the gene expression profile to understand something about what the cell actually is capable of doing beyond just what's encoded in its genome. Another layer of phenotype is the proteome. And the challenge with the proteome, unlike the transcriptome, which is a nucleic acid, right? So you can sequence the transcriptome using DNA sequencing, and you can measure phenotype that way. But the proteome is qualitatively different because it's comprised of proteins, and sequencers don't sequence proteins; they sequence nucleic acids. So back then we said, well, is there a way we could somehow integrate that protein information into something we could sequence? And that became the development of what we called AbSeq, where the idea was to build a molecule that was a conjugate between an antibody and a nucleic acid. Essentially, what we do is we take our cocktail of antibodies that you would typically use for FACS or just immunohistostaining, and rather than labeling them with fluorescent dyes, we label them with oligos, with specifically defined oligo sequences. So each antibody in my cocktail gets a known tag sequence. Now, when I hybridize, or when I bind those antibodies to my cells, they're going to bring with them these foreign DNA molecules that essentially tell us that they're present. So now I've essentially transduced a protein signal into a nucleic acid signal that I can measure by sequencing. And that became what we called AbSeq, and published it in 2017. And then about a year after that, some clever labs realized, well, you could now just do AbSeq together with RNA-seq if you just polyadenylate those tags. And so they used AbSeq together with RNA-seq, and those are called CiteSeq and ReapSeq, respectively. And these are beautiful technologies. They are really the first to show the potential to do really broad kind of multi-omic analysis at the single cell level. But there is, I would say, a limitation with CiteSeq and ReapSeq, which is that, again, you're measuring RNA and protein, and in some sense, those are both measurements of phenotype. What you really want is to be able to link genotypes to phenotypes. And so my lab and Ben and Cyrille, who are the developers of the technology, developed a new technology, which we call DABSeq, which is now multiplexing the DNA genotyping of Tapestri with our AbSeq technology. With that, I'll let Ben and Cyrille take it away.
Thank you, Adam. So I go next, I guess. So what we felt like, if you have a cancer sample or a tumor sample, two ways to analyze those kind of samples. So typically, either you do DNA genotyping and you look for the mutations that are harbored within this tumor cell population, or you use flow cytometry to look at the marker profiles of the cells. And those are used for now decades in cancer research. But everybody knows basically that the tumors are genotypically heterogeneous, and they also can be very heterogeneous in their marker expression. But we wondered, how do they really connect? And we feel like you can only know the relation of the phenotype to the genotype if you measure them together on the same cell. That's why we developed basically the AbSeq technology. So Ben and I went ahead, next slide, Ben, and basically designed the workflow based on the Tapestri device from Mission Bio, which makes it very practical. So now the workflow is really very easy to be employed. We basically start by taking a patient sample and our oligo tagged antibodies, and we stain the cells from the patient sample just like you would do for a FACS experiment. But instead of running it through the flow cytometer, we basically move to the Tapestri, or like to its microfluidics, and encapsulate those cells into the lysis droplets, where the cells are lysed, which liberates the genome and also degrades the antibodies. And in the second Tapestri step, those cell lysates are barcoded with cell-specific barcodes. And in this, it's basically droplet PCR. What we are doing, this reaction attaches then the cell barcode to both genomic loci of the cancer targets you are interested in, and also to the phenotype, basically to the handles of those antibody barcodes. Next slide. And so for our study, we decided to look at blood and at leukemia in particular. So therefore, we designed an antibody panel of the typical hematopoietic cell markers. So we used 23 different antibodies simultaneously, and at the same time, we had a DNA panel comprising 50 targets, so 50 amplicons spread over 19 genes. And next slide. So to test the system, we went ahead and used a PBMC sample from a healthy donor and stained it according to our workflow, run through the Tapestri workflow and sequenced the antibody library. We didn't sequence the DNA library in this experiment because we don't expect to find anything interesting in a healthy blood sample. So after sequencing, we get antibody tag counts based on the UMIs in the antibody tags. And we need to correct those counts, those raw counts, for different efficiencies of the droplet amplification process. So we use a linear regression like people do in the field of RNA-seq to correct the count numbers. And then we visualize that in UMAP representation, as you would also see typically for RNA measurements. And so that's our UMAP for the PBMC sample. You can see it's kind of quite complex. This UMAP sample is so just to remind you, it's based on those 23 antibody counts. So we have only like the 23-dimensional vector is underlying this space. So it's much smaller than what you would see in a typical RNA-seq experiment where you have something like 20,000. But since those 23 are basically cherry-picked, we get a very fine-grained resolution on the different compartments in the PBMC sample. And you can like zoom in basically at the bottom right. We have, for example, the T-cell compartment, which has, for example, CD3 expression, or on the left side, we have the B-cell compartment, CD19, CD20 expression. So we can basically annotate those different cell compartments. That I hand it off to Ben.
Okay, thank you, Cyrille, for that great explanation of the validation experiment. So I'm going to talk about a couple of the different patient cases we featured in our most recent publication, which again is available on bioRxiv. If you want to find it, I think it's easy enough just to search on Abate and it should show up as one of the first hits. So if I run through some of the figures, you can always consult the publication for more details. I want to briefly mention, of course, from the onset that this was a collaboration with Catherine Smith at UCSF. She's a hematologist there, along with Cheryl, who is one of the great hemoncs in her lab. And it was through them that we actually got these patient studies or patient samples, and we were able to process these in a way that made sense for us. So the first patient I want to profile is one where we see a very clear correspondence between the immunophenotype of the blasts, which are these malignant cells, the leukemic cells, across different therapy time points. And one other thing I want to mention is that I thought our studies that we profiled in this paper were very good at showing the response of cells over the course of treatment at multiple time points, 4 to be exact in this case. And I think that's something that's rarely seen in RNA-seq papers, for example. Usually you look at 1, maybe 2 time points. So I think this does give a good sense of how these cells behave across therapies and in response to different therapies, albeit with a small cohort. So patient 1 was a 47-year-old male diagnosed with AML. This particular patient had a very extensive treatment history. I've only summarized that here on the left side. Mutations in these particular blasts includes one in a DNA methyltransferase, DNMT3A, and then also an NPM1, which are present at this relapse time point, which was actually not the first time this patient had relapsed. In the final time point, or prior to this remission time point, this patient was treated with a CD33 drug conjugate, gemtuzumab. CD33 is a pan-myeloid marker, so this drug conjugate is intended to act against cells of a myeloid lineage, so the blasts in this case. And this patient did receive complete remission after this time point. Looking at the clonality of these cells in terms of their genotype, which is what we get from the DNA side of things, we essentially see that there's 1 of 2 genotypes in this sample. It's a healthy cell, or at least wild type across all the different targets we profiled, and then 1 that is co-mutated at both the DNMT3A locus and NPM1 locus. So one of the cool things I think about essentially what expression of one antibody versus another, and that's just and that's just a conventional 2D scatter plot. But in this case, with the DAB-seq data, we're able to overlay this with where these mutated cells lie on this plot. And in this particular instance, I wanted to kind of show this concisely by looking at 2 markers people look at frequently in AML, CD33, which is again the target for the gemtuzumab, the pan-myeloid marker, and then CD34, which is used to subtype certain types of AML. And essentially what we see here, and I've plotted this not as a scatter plot, but as a contour plot. The data type is essentially the same, though. We see where the densities of cells lie, meaning, for example, if I look at this first time point, the second relapse time point, I see that this patient has many cells, both in this compartment that is CD34 positive and CD33 positive. Not only are there a lot of cells, but there are a lot of NPM1 mutated cells, which I highlight with the red contour. So rather than just seeing, you know, this cell is CD33 positive or this cell is CD34 positive, I see that these cells are also mutated. And how, and you know, and obviously at what locus as well. That's even richer information that I didn't show on this plot. So for these particular clones, we see across treatment time points that they maintain a fairly robust immunophenotype, and by that I mean that they don't really change the expression of CD34 across time. Their numbers may change, although the relative expression between, say, CD34 positive and CD33 negative cells doesn't change. And we do finally see at remission that there is an absence of mutated cells, and that's obviously a good thing to see. Nor do we see that the CD33 negative, CD34 negative quadrant here in the lower left has any mutated cells at all, which is sort of a good validation of the technology. So while this may be a simple case, it does demonstrate the power of the technology to kind of combine conventional flow cytometry data with genotyping data. This next patient, which will be the second and last one I profile here, is a very interesting case, in a case in which we showed that this FLT3 inhibitor, gilteritinib, induced this differentiation response in the myeloid blast of a patient diagnosed with AML. So this patient, again, is the second patient, 59-year-old female diagnosed with AML. Similar to the last patient, there are mutations discovered in DNMT3A and NPM1, but in this case, we actually see at the third time point, which we label relapse, there's the we discover a 24 base pair FLT3 internal tandem duplication, ITD for short. So basically, what this looks like on the DNA type of things is that we see that these cells have they have either DNMT3A mutations, or they're also co-mutated at the NPM1 locus and the FLT3 ITD locus. So it's essentially accumulating a series of mutations across time. These cells are, and it's interesting to note that, you know, gilteritinib is advised for use with FLT3 ITD patients, though the differentiation response of these cells in response to that gilteritinib it remains to be fully understood, I think. And so one of the ways that we can use this technology is to apply it directly to the question of, okay, for all of the clones that contain this ITD, what is their phenotype now? What is their lineage within the blood cell compartment? Are they still myeloid blasts? Do they still look like myeloid blasts, or are they differentiating into another cell type? And this is really a question that can only be directly answered with something like this. So again, as Cyrille described previously, we're able to take this high dimensional data, which is the counts of all 24 of our antibodies, and plot these as a 2D visualization using the UMAP algorithm. And so we do that for all the time points together, and we get something like 18,000 cells. When we cluster these cells and annotate them based on distinctive markers within each cluster, so for example, the CD3 positive lymphocytic population or the CD71 positive erythrocytic population, we see that there's these constitutive markers that show up very clearly in there. But again, this is from all of the time points. This UMAP is from all the time points combined. So we want to understand now, by looking at the time points individually, what happened in between these therapies, and how did these cells propagate between clusters that were, you know, either one of the diseased blast clusters or something that looked more like a healthy erythroid or lymphoid cell. And so this is the last figure of our paper, and it's a little bit dense to show on a slide here. So I urge you to consult the paper for a full understanding of it. But essentially what we've done is, across each of these 4 time points, we've plotted the same UMAP I showed in the previous slide, and then overlaid this with colors corresponding to the genotype of each of these cells. Blue meaning the wild type, with this sort of darkening shade of red corresponding to 1, 2, or 3 co-mutated loci. Along with that, we've plotted these genotypes, again, the CD33 expression. We use CD33 expression essentially as a proxy of whether these cells are myeloid or not, because we know that, again, that this is a pan-myeloid marker. So we see that, unsurprisingly, cells mutated at the NPM1 locus have a high degree of CD33 expression. Similarly, we can take the distribution of these cells across each of the clusters and plot this here in this log scale chart at the bottom. And so at diagnosis, we see that most of the cells are concentrated very heavily in these BLAST1 and BLAST2 clusters, as well as the BLAST3 cluster. There are some healthy monocytic cells in there as well. At remission, we observe that most of the cells have a wild type genotype, meaning they're not mutated at any of these loci that we've looked at. However, we do find that there's a small amount mutated at DNMT3A. That in itself, I don't think would be too alarming for a clinician, although it may raise some eyebrows. I think that this is clear evidence that because CD33 expression is low, those blasts may not pose an imminent threat to the patient. And this here at relapse is when we find that this new clone with the FLT3 ITD really emerges with a very high CD33 expression level, and unsurprisingly, these are all myeloid blasts. And now for this last time point here, where this patient is treated with gilteritinib, we see that most of the cells, the vast majority, in fact, still contain this FLT3 ITD clone. But now the CD33 expression is vastly reduced. Not only is it vastly reduced, but we have this corresponding shift to this cluster here in the lower right. That's the erythroid cluster I spoke about in the previous slide. So essentially what we infer to be happening here is these cells, these previously myeloid blast cells with the FLT3 ITD mutation, have propagated from a myeloid phenotype, a myeloid blast phenotype, into a erythroid phenotype, and we take that to mean that they've differentiated in response to the gilteritinib. And consulting with Kathy Smith, the hematologist at UCSF, this makes a lot of sense, and we found some recent work in the field that kind of validates this conclusion. And this is just a figure I pulled from that paper supporting what we saw as well, which was that patients on gilteritinib, they have this erythroid differentiation response to that drug. And so I think what we're able to do here with Doubleclick is very concisely show by providing the direct evidence, measuring DNA and phenotype within the same cell, that this is indeed the case, at least for this particular patient. So this phenomenon could certainly be studied on a wider scale, but I think what we've done here is a very nice result. And hopefully should inspire other clinicians who may be seeking similar answers to what might be going on in these sorts of cases. And so that's going to wrap it up for this second patient and for the presentation. Again, I urge you to look at the paper if you want to know more, but here's our list of acknowledgements. One other person we definitely want to give a shout out to is Harish in our lab. He's a radiation oncologist who really also did some great work for the paper by consulting about some of the clinical aspects of it. All the people in the Smith lab, of course, and then Mission Bio too was obviously instrumental in getting this off the ground. So with that, I guess we open it up to questions. Thanks, everyone.
Thank you so much, Ben, Cyrille, and Adam. That was really wonderful. So we have a few questions here. So a couple of technical questions. Someone wanted to know, did you validate the data facts from the same sample?
Say that again. I dropped out halfway through, please.
So this attendee wanted to know, did you validate the data with facts from the same sample?
We didn't show it here in the presentation, but we did some cell line tests up front, and we validated those with facts. Yeah.
Yeah, the PBMCs we also with the smaller subset of the panel we did validate that using multicolor flow. Yeah, and it looked very good.
Great. I'm just curious, were you able to do all 23 antibodies on a FACS instrument?
That's difficult even for an expert operator, and it would probably just require multiple tubes. So the answer, unfortunately, is no. I think we did like 6 or 7 at once, which already itself requires a lot of compensation. You know, I think this sort of speaks to the simplicity with which you can encode stuff in DNA tags, and as the field moves towards this, they'll become a little bit more trusting of the sorts of data we get.
Definitely. So along that same line, how big of a panel do you think you can do for the protein and then also for the DNA?
Well, I think if the papers coming out of other groups who do this sort of thing is any evidence that there's really no limit to how much you can multiplex the antibodies as long as you're willing to spend money on sequencing and the conjugation. So it could be a couple hundred, or more. Whether or not all those markers are useful and relevant to you is another question.
That's certainly really exciting. So did you sequence the protein and the DNA together?
Do you want to take this, Cyrille?
Well, yeah, in some sense. So we prepare the libraries independent of each other after the Tapestri workflow. Ultimately, depending on the sequencing technology we use, sometimes we sequence it together with different indices, or we sequence it on different lines. Usually it's advisable to use different lanes if you have enough sample, if you can basically cover it with enough sample material, but if not, you cannot totally sequence them together in one go.
Great, thank you. And then I'm curious, I know you collaborated with Kathy Smith looking at patient samples from AML. What are other good indications or areas of research that you're excited to use this technology to look at genotype plus phenotype?
Do you want to take this, Cyrille?
Well, I guess the obvious answer, everybody's very excited about solid tumors, besides leukemia type of tumors. So that's something we are actively looking at now.
Yeah, I would add on to that I think that's the next frontier for a lot of single cell stuff. Hematology is great, but it's it's very convenient because you have a single cell suspension already in the blood. But I think that there's a lot of cool stuff that can be learned from solid tumors, and Harish here in the lab is really helping us move that forward.
That's certainly very exciting. So do you think you could get away with still doing surface protein expression for a solid tumor, or would you want to move into intracellular proteins?
That definitely cuts to the heart of the problem with solid tumors, that you always have the trouble with the dissociation. It really depends on how successful you are with dissociation, I think, if you can look at surface proteins or not. But we will probably know more within a couple of weeks, a month.
All right. Well, with that, that's a really great teaser. We would love to have you back in a few weeks or months to talk about your next set of experiments. But really, we really appreciate the Abate Lab taking the time out to share with you their new paper on DNA plus protein in bioarchives. And thank you so much for joining us at home. We hope everyone's doing well.
Thank you.
Thanks, Kelly.